Hello students, I welcome you all to KK's biology class. What are we studying today? We are studying the second part of our discussion, that is, what is staining. So what is staining, students? Staining is a procedure wherein I am going to be making use of stains. What are stain? Stain is defined as an organic compound. An organic compound which is made up of two important components, one being a chromogen and second being an oxochrome. If any organic compound has to be called as a stain, it needs to have both of them, that is oxochrome as well as your chromogen. So what are these oxochromes and what are the chromogen? Chromogen is in turn made up of two different parts, one being benzene and the second one being chromophore. Benzene, as all of us know, this is a colorless solvent. It's an organic colorless solvent. The second part of the chromogen is interesting. Why? Because it is made up of chromophore. And what is a chromophore? Chromophore is that chemical group which is going to be imparting color to benzene. Meaning what? This chromophore has the ability to absorb the radiation anywhere between 400 to 800 nanometers, which we call it as your visible spectrum. But chromogen is a colored compound and not a stain. Why? Because chromogen alone cannot have the ability, it does not have the ability or does not have any affinity to bind with fibers or tissue. Therefore, for any organic compound to be called as stain, as we said, there are two parts, chromogen and oxochrome, and oxochrome is also required. So what is this oxochrome? Oxochrome again is a chemical group. What is this chemical group doing? This chemical group is conveying the property of ionization. We also call it as electrolytic dissociation. What is electrolytic dissociation and ionization going to be doing? It is going to enable to form salts and thereby binding to the fibers and tissues. Very essential that these oxochromes have to be present. So, revising again, what did we study today? That what is a stain? And stain is nothing but stain is going to be A plus C. What is A plus C? A is the oxochrome and C is the chromogen. Chromogen in turn is made up of two parts. Which are those two parts? One, chromophore and benzene. Chromogen alone cannot be called as a stain. Why? Because it does not have the affinity to bind to any fiber or tissue. Therefore, an oxochrome group is required. What is oxochrome group going to be doing? It is going to be conveying the property of ionization or electrolytic dissociation to the chromogen, which thereby will enable it to form and salts as well as bind to the fibers or the tissues. Thank you students. This is what for today. Do write us to kk'sbioacademy@gmail.com and do not forget to subscribe my channel and hit the bell icon. You can also scan the QR code to book for a learning session. Thank you.